All right, I've got 501 here, so I think we'll get going. Good evening. It's my pleasure to welcome you all to the second installment of the Community Science and Technology Special Virtual Seminar Series, Demystifying COVID-19, presented by Pacific Northwest National Laboratory. I hope you were all able to join us last week for our first talk. If so, welcome back. If not, thank you for joining us tonight. As was the case last week, this session will be recorded so that those who are not able to join us today can view it at a later date. My name is Jim Sanford. I'll be your moderator again today as we hear the second presentation of this exciting 5-part series designed to share what we've learned and answer some questions related to the global coronavirus pandemic that has affected us all in various ways over the past year. Before we get started, we'd like to take a quick moment, as we did last week, to see where people are joining us from. There will be a link provided in the chat window. If you could click on that or follow the instructions click on that link in the chat window from your computer or mobile device, and it will take you to our polling feature. And we should be able to have some live results popping up on the screen here in just a moment. Here we go. Okay, this is what we were trying to accomplish last week. We got a nice map and a little word cloud showing up of where everyone's tuning in from. Good Tri-Cities and Washington turnout, but do see some folks from a little further away. That's awesome. Cool. This is quite nice. People from all over the country. I think even some from abroad. Wonderful. Okay, well, thank you everybody for joining us tonight. Thanks for participating in that poll. It's wonderful to see such a nice turnout. We'll hop back in here to the presentation. I don't want to take up too much time because last week the question and answer session was my favorite part, so I want to leave as much towards the end of that. But did just want to share a couple opening notes about Battelle and Pacific Northwest National Lab. PNNL is one of 17 United States Department of Energy national laboratories, and we're focused primarily on addressing critical national needs and advancing the DOE's missions in some of the scientific areas demonstrated here on this slide. On the next slide here, we have a few metrics indicating some of the economic impacts that PNNL has on communities across Washington State and beyond. Our primary campus is in Richland, but we do have locations in Sequim, Seattle, Portland, Maryland, and Washington, D.C. Besides an economic impact, PNNL strives to be good stewards in the communities in which we live and work. Battelle has invested millions of dollars, and PNNL employees log thousands of volunteer hours each year with local organizations and through outreach activities. One of our mechanisms of outreach is this Community Science and Technology Seminar Series. A link at the bottom of the page here will take you to a PNNL events webpage where you could find information about future upcoming seminars. Typically, these community science and technology seminars are talks given by PNNL scientists focusing on the research being conducted at PNNL. But this particular case, we decided to try something a little bit different. As we're illustrating here, we put together a 5-part seminar series focused on various aspects of COVID-19 science. We have 5 PNNL researchers who have volunteered to give presentations on the indicated topics. Each talk is beneficial on its own, but the seminar series is designed to complement and build upon itself, so we strongly encourage you to attend all 5. As an added bonus, those attendees who complete the survey provided at the end of each talk will receive a digital certificate of completion. So last week, the seminar series started off with a great talk by Wylie. Again, I hope everyone is able to tune in for that. If not, the talk has been recorded and posted on the PNNL website, so you could always jump back and find that. Tonight, we're going to be hearing from Amy Sims, who will be giving an overview on what we know about the origins of COVID-19 and other viral diseases. I'll introduce Amy in just a moment here, but first I wanted to remind everyone or introduce those just tuning in for the first time about the format of these presentations. Today, Amy will give a 25 to 30-minute presentation, after which we'll have time for question and answers. I encourage you all to type your questions into the Zoom chat as we go along. Those are being collected and we'll present those to the speaker at the end as time allows. To help spur the question and answer session at the end, we are joined again by a wonderful panel of community representatives from various organizations, and they have come prepared with some questions related to the interests that they represent. So allow me a moment to introduce our panel again this week. First, I'm going to be going from right to left today, just so we can shake it up a bit. First, we have Martín Valadez, the Regional Director for Heritage University's Tri-Cities campus and the Interim Executive Director for the Tri-Cities Hispanic Chamber of Commerce. Martín, what are you hoping to learn tonight?
More about the connection between, how infections get carried from non-humans to humans. And then I can tell something to my son that he doesn't know about bats.
Perfect. That's great. Next, we have Justin Rafa, the Artistic Director of the Mid-Columbia Master Singers. Welcome, Justin. A few remarks, please.
Thank you, Jim. I've come to consider the loss of taste and smell among the main indicators of whether a person has contracted COVID. And as someone who finds great joy in life from eating and drinking, I wonder if I have placed too much emphasis on this factor, but I'm also eager to learn more about why this unusual symptom occurs.
Excellent. Looking forward to Amy enlightening you on that. Next, we have Kate McAteer, the Vice Chancellor for Academic and Student Affairs at Washington State University Tri-Cities. Welcome back, Kate.
Thank you, Jim. I'm excited to be here and for this talk tonight. And I'm interested in learning more about how the immune system of these small creatures can evade illness, yet we humans, which might be considered as having more complex immune systems, get ill from these same viruses.
Excellent. And finally, rounding out our panel today, we have Loanne Ayers, the President and Chief Executive Officer for United Way of Benton and Franklin Counties. Loanne, welcome back. What do you hope to gain from today?
Well, thank you, Jim. Many of the employees, volunteers, and clients of local nonprofits traveled frequently to visit family out of country in pre-pandemic times. And when travel reopens, there's a lot of anxiousness about reconnecting. There's eagerness to do so, but caution about what's safe and how can we prevent future outbreaks as we reconnect with our families in other countries. So that's what I'm interested in learning more about.
Awesome. Thank you very much. Thank you all for your attendance tonight, for those thoughtful remarks. Without further ado, it gives me great pleasure to introduce Amy Sims. Dr. Amy Sims is a virologist at PNNL who studies how the DNA manipulation strategies of viral attacks widens the path for future therapies. She earned her PhD at Vanderbilt University Medical Center and spent 15 years at the University of North Carolina, Chapel Hill before recently coming to PNNL. Amy, welcome. Take it away.
Sorry about that. Jim was a little quicker than I was anticipating him being, so I didn't get myself unmuted, and I'm hoping that someone will tell me if you cannot hear me. All right. So I'm going to assume that you can hear me until someone tells me otherwise. So I really hope, as Jim said, that you guys got to hear Dr. Wylie's presentation last week. It was really fantastic. It was a great overview of COVID-19 and some of the kind of general things that we've learned overall. Today, in the next couple of weeks, we're going to focus in a little bit more. Today, I'm going to focus in a bit on the transmission portion of the viral infection process, talk a little bit about bats, as well as some general things about SARS coronavirus 2 and the disease of COVID-19, as well as some potential treatment options and where we go from here. So one thing I definitely want to do is to make sure that we're all on the same page and that I'm not using words that are confusing. Definitely put that in the chat if I say something that you're not sure of. I want to make sure that we all leave here with good information. So this is kind of a terminology slide. If you're with us last week, Steve went through the same thing, but we're going to do this each week just to make sure everyone's on the same page. So in a pandemic is an outbreak of a disease that is prevalent over a continent or the world. Note that an epidemic is also an outbreak, but it's a little bit more localized. So when I say transmission, we're basically just talking about spreading of a disease. Mutation are changes to the genome or nucleic acid of a pathogen or organism that may affect transmission, symptoms, or prior immunity. When I'm talking about being infectious or contagious, this is basically the state of being able to transmit a disease to another person. So vaccines are a preventative measure to build immunity against a specific disease. A model is a representation of a disease or process that can recapitulate key aspects. And a reservoir is any person, animal, plant, soil, or substance in which an infectious agent normally multiplies or can kind of hang out for a long period of time. And so, as Jim mentioned, I am a virologist, and so which means I study viruses for a living. And so I tend to use COVID-19 and SARS coronavirus 2 with slightly different meanings, and so I want to make sure that I'm clear about what I'm saying. So severe acute respiratory syndrome coronavirus, or SARS coronavirus 2, is the literal virus that causes COVID-19. And COVID-19, or coronavirus disease 2019, was named by the World Health Organization, or WHO, and it's basically the potentially severe respiratory infection that's caused by SARS coronavirus 2 virus. So the general overview of my talk, I'll talk some about virus replication and disease outcomes in animal hosts and humans, talk a little bit about what we knew about human coronaviruses prior to the global pandemic of COVID-19, talk about COVID-19 disease symptoms and SARS coronavirus 2 transmission, and spend a little bit of time talking about the loss of smell and taste, which should be good. Then move on to SARS coronavirus 2 genome changes over the course of the pandemic. What are current and future COVID-19 treatment options, and then what happens next? How do we prepare for the future? So I'm going to start talking about viral replication and disease outcomes in animal hosts and humans. So we knew before SARS coronavirus 2 that coronaviruses could emerge or move from animals to humans or animals to animals. And coronaviruses actually infect a very wide range of animal species. And I'm showing some of them here as kind of silhouettes on this slide. So you can see that coronaviruses can infect everything from humans to bats. You probably had your dog vaccinated against canine coronavirus, but it can also infect things like seals, whales, a very, very wide range of animals that the viruses can infect. And like I said, we've known about animal to human coronavirus transmission. It's actually been occurring for thousands of years. So we know in some previous studies that the human coronavirus NL63, which causes kind of a mild cold to bronchitis, actually made a transition, we think, from bats to humans in about 1100. So, you know, a very long time ago. Whereas the common cold-causing coronavirus, OC43, which we think started off as actually a bovine or cow coronavirus, transitioned to humans in the 1800s. So this has been happening for a very long time. So SARS coronavirus 2 is not the first time. But it definitely has made a very lasting impression on the world at large. And so, you know, as you probably are aware that as humans and animal habitats overlap or where they live, you know, as humans take more space for living and take more space away from animals, that we're going to come into closer and closer contact with them. And it really makes transition events become more likely. So I think that's something that we as a, you know, as responsible citizens of the globe have to figure out how to have you know, more safe interactions or, you know, limit interactions with animals that could potentially lead to different transmission events. So, you know, the bat has definitely become kind of like a superstar in this particular pandemic as, you know, kind of the main reservoir of SARS coronavirus 2. We're still learning, you know, the virus, we're still learning about this virus. So we don't have everything figured out yet as scientists. And so we're still trying to do experiments to understand exactly how the virus made the jump. We think from the bat reservoir into humans, maybe there was an intermediate animal with an earlier coronavirus. We think that the animal reservoir of camels contracted it from the bat, then passed it to humans. So we're still trying to figure all that out for SARS coronavirus 2. There's hints that maybe it was the pangolin. But the reality is, you know, bats that are infected with these coronaviruses, they don't actually get sick. They don't have any disease symptoms, so it's not like you can identify that bat, likely has a coronavirus, and maybe we need to try to do something about that one. But the reality is they don't have any disease symptoms. And current studies suggest that bats have a reduced immune response kind of overall. And the current thinking is that it has something to do with being the only flying mammal, that there's some kind of link between that. And I'm not exactly sure what that is, and they're still doing studies to figure that out. But we think that somehow the fact that the bats fly have contributed to having a reduced immune response. And this in turn makes them kind of an ideal animal reservoir for many different viruses. However, I also do want to stress that bats are a really, really critical portion of the ecosystem. I come from the deep south, and we want to make sure they eat all the mosquitoes that they like, because obviously that's another critter that we don't want around. And so just because bats seem to harbor these viruses does not mean that we need to wipe out all the bats. They're an extremely important portion of that ecosystem. All right, so what do we still have to learn about transmission and how viruses move from animals to humans? So that's actually something that we definitely want to understand, is how these transmission events occur, what would be indicators of those transmission events, and why is it that some viruses seem to require an additional animal species before moving into humans. We also want to learn how to prevent transmission events from taking place in the first place. That would be fantastic, and so how do we figure that out? So scientists are working really hard and trying to figure out how they can make that, what changes are necessary in the virus to be able to move from an animal to a human. We also want to figure out how to encourage public health measures, potentially different hygiene, different perspectives of hygiene that could also prevent future outbreaks. If you're handling these types of animals, lots of hand washing, things along that line, to try to prevent any sort of future outbreaks if we possibly can. So next I want to move to talking about human coronaviruses and what we knew before the global pandemic. So just kind of a timeline of human coronavirus identification is what I'm showing here. So unlike influenza virus, which we've known about for more than 100 years, the first human coronavirus, which is actually a cause of the common cold, wasn't really identified until 1960 to 1970. So we've really only been working with these viruses for a relatively short period of time, so we still have lots to learn. It turns out that these common cold-causing coronaviruses are relatively difficult to work with. And so most of the work that's been done and most of the things that we've learned about how coronaviruses copy themselves and lots of different general information about viruses have really been done with different model viruses, including the mouse hepatitis virus, which, as the name suggests, infects mice. Coronaviruses are actually very important domestic animal pathogens. So they infect chickens and pigs, and they can really wipe out entire herds or flocks. So we've learned a lot about coronaviruses by studying those. And really until about 2003, people didn't study human coronaviruses very much at all. The big thing about 2003 is then that's when severe acute respiratory syndrome coronavirus the first, or SARS coronavirus the first now, was identified, which was really a huge change for the field. People really thought that human coronaviruses only caused the common cold, and so that caused a major shift in a lot of people to move into the field. And so to date, 7 human coronaviruses have been identified. After SARS coronavirus the first was found, people began to go back through clinical samples that they had, and they identified 2 additional less severe human coronaviruses called HKU1 and NL63, which cause kind of a bronchitis or a kind of a severe cold. And so there was a lot of thought till about 10 years after SARS coronavirus the first was identified that maybe this was going to be the only one. We weren't ever going to see it again, although some of us who were in the field were kind of predicting that that was less likely to be the case. And lo and behold, in about 2013, the Middle East respiratory syndrome coronavirus, or MERS, was identified. And then, as everyone knows, 2019, 2020, SARS coronavirus 2. So a little bit more information about the epidemic of SARS coronavirus 1. It caused about 8,000 cases and about 800 deaths, which is about a 10% mortality rate. This virus is no longer considered to be circulating, and the epidemic caused by this virus lasted about 8 months total. So you can no longer contract SARS coronavirus the first out in the world. For MERS, since about 2013, this virus has caused about 2,500 cases overall, with about 850 deaths. This is about a 34% mortality rate, which is very, very high, obviously. And we think this number may be slightly high because it is likely that there's a large number of asymptomatic cases of MERS coronavirus that obviously would not necessarily be reported because of the close interface between humans and their camels in the Middle East, because camels can be for food, they can be pets. You know, there's a lot of close interface, and so we think that this virus is definitely still spreading. There's still sporadic cases that are going on, so this is actually something that you can still contract. So just so that you have kind of a feel for it, you know, we've really only known about these viruses for a little over 50 years or so. So we're still definitely very much in a learning phase. But the good news is, and something that I try to express to my mom on a frequent basis, is that the basic research that we had done in the previous 20 years with SARS coronavirus the first and MERS taught us and scientists in general what the most immunogenic, or what the best protein of the virus was for generating an immune response. And that's actually one of the things that allowed the vaccine manufacturers to identify a target so quickly and to get on making a vaccine as quickly as they did, is that we had this basic research already completed. So just to kind of put that out there, you know, you never know where your basic research is going to take you. So please keep supporting the National Institutes of Health and national laboratories as well. So next, I want to talk a little bit about COVID-19 disease symptoms and SARS coronavirus 2 transmission. So SARS coronavirus 2 actually infects many areas in the respiratory tract. And so this is in slight contrast to MERS and SARS coronavirus the first, which when we looked in patients, replicate really primarily deep within the human lung. So what I'm showing here is that the virus can replicate either in the lower airway, or what I've termed here is the parenchyma or the alveolar sacs, where the air exchange takes place. So basically, to spread MERS or SARS coronavirus the first, it really required, you know, close contact with the patient and a deep lung cough to be able to get the virus out and into a breathing space where someone else could breathe it in. This is in contrast to SARS coronavirus 2, which actually seems to replicate in several regions of the respiratory tract, not just deep in the lung. So this includes like the linings of the nasal cavity, as well as the conducting airway, and deeper into the lung as well. So because of this, you know, transmission of this virus, you basically just need to speak or talk or cough or laugh, you know, or these sorts of things. So it's much easier for SARS coronavirus 2 to spread to other people. So where the virus replicates in the lung can change, you know, transmission rates, and so it can facilitate transmission. But in another way, SARS coronavirus 2 being in the nasal cavity actually makes it easier to do the diagnostic test, rather than having to get a deep lung sample, which made MERS and SARS coronavirus the first actually much harder to diagnose. So if you do actually get infected, as you probably all know now, what do the infection symptoms and disease severity look like? So you're probably looking at flu-like symptoms, with the most likely loss of smell or taste being very common. It's actually also possible to have nausea, diarrhea, or skin rash following infection. And I think Dr. Waters will talk a little bit more about this next week. Severe disease progression includes potentially going into acute respiratory distress syndrome, or ARDS. Can also include neurological complications, kidney injury, shock, and potentially multi-organ failure and possibly death. As with one of the community leaders, I've also been really fascinated by this loss of smell and taste as a symptom of being infected with SARS coronavirus 2. And I guess in my head, I always kind of think about this as kind of having like a stuffy nose and just not being able to breathe, and that affecting your ability to taste. However, you know, when I was doing a little bit more investigating about it, I actually found that infection with a range of other respiratory viruses that can replicate in the upper airways and nasal cavity can also result in this loss of smell or taste, but usually with a lot less frequency than is being seen with COVID-19. And so right now it's still actually unclear, and scientists are trying to figure out if this loss of smell and taste is a result of some kind of inflammation in the nasal cavity, or if it's actually infection of the olfactory sensory neurons, as I'm proposing over here. And so what do I mean by that? So basically, the virus can actually infect the cells that are lining the nasal cavity, as I mentioned before, and it actually prefers to infect these guys shown here in yellow with kind of like the little hair or cilia on top. And these cells are actually right next to the olfactory epithelium. And so these are the cells. Within these cells are contained the neurons that would be responsible for smell and taste. So the virus might actually be able to infect some of these other cells, and causing cell death or damage to this tissue could result in this loss of smell or taste. And so there are actually ongoing experiments trying to better understand this. But we think that's, one of the possibilities of what might be going on here. So I just wanted to cover that with you because that's something that's been of great interest to me as I'm learning more and more about this virus. So what do we still have to learn about this part? So what we found is that adults with pre-existing medical conditions have more severe disease outcomes, and we still don't really understand why, all those reasons why. Something else that's been very interesting about this pandemic is that typically with SARS coronavirus, the first, or MERS, we saw little to no infection in children. But again, we had a much smaller number of people who were infected. With SARS coronavirus 2, we're seeing this multisystem inflammatory syndrome in children. I looked on the CDC website today, and there have currently been over 2,000 cases identified with this syndrome in children. And so we're still not sure what's going on. I don't think that there are treatment options yet, but that's definitely a big area of research interest. And so people are trying to figure that out. But that's actually one thing that we really want to learn more about why that's happening. With SARS coronavirus, the first, and MERS, as well as SARS coronavirus 2, the elderly seem to have much more severe disease outcomes. That's something that we're still trying to work on in aged models and trying to figure out what it is about the older immune response that tends to lead to more severe disease outcomes. And then finally, we're trying to figure out more, does blood type influence either your ability to get infected or possibly disease outcomes? I think there's more research showing now that it does not necessarily affect disease outcomes, but may affect how readily you can become infected. So now we're going to switch gears and talk about SARS coronavirus genome changes over the course of the pandemic and the different mutants that we're seeing. So viruses, unlike the cells in your body, actually don't do a very good job of making copies of themselves. And so, when you have a cell in your body, it wants to do the best job possible to make an exact clonal representation of itself so that your body continues to function as you need it to. Viruses don't seem to have to care about that because they can make a lot of progeny, lots and lots of baby copies of themselves. And so the viral enzymes that make the copies of the genome with nucleic acid inside the virions, they actually make mistakes each time that the genome is copied. And this can actually result in very large mutant populations. So you don't get infected with one virus and then have a million copies of that virus come out of your body or your cells. You actually have kind of a ranging population of viruses that come out. And so animal hosts or reservoirs where the virus can replicate without an effective immune response allow for a much larger mutant population to develop. Now, it turns out these mutations can actually be either beneficial or harmful to the virus. The mutations are random, so the virus doesn't actually control where these mutations are happening. So it could be that, all but one virus that comes out of a cell has all deleterious or bad mutations, and only one virus can move on. And so that's just kind of one of those things, and the virus makes enough copies of itself that it's willing to kind of take that risk, if you will. So here in the schematic, I'm basically just showing you that the mutations that kind of pop up are all throughout the genome, and they can occur anywhere, and there's no real rhyme or reason to it. So why is SARS coronavirus 2 changing over time? Well, it too has this, error-prone or, enzyme that doesn't do a very good job making exact copies of the genome. And so it's making mistakes in the same way. And basically, because it's been able to infect so many people, we're seeing the virus do a better job of being able to transmit from person to person. Obviously, as you learned last week with Steve, that viruses require a host to replicate. And because these viruses make mistakes each time they replicate, they can actually potentially generate mutants that can adapt to the host or do a better job of moving more quickly through a host or being more transmittable. And so I think that's basically what we're seeing now, is that some of these mutants can just move between people faster, so that these viruses have an advantage. And so that's what you're starting to see. So scientists are still very actively learning more about these viruses as the mutants are identified. So we're still trying to figure out what all this means as far as how the mutants are contributing overall to the pandemic. So now I want to move to talking about current and future COVID-19 treatment options. So I want to kind of highlight three different COVID-19 treatment options. The first being antivirals. So obviously virus particles multiply inside of your body. And so we basically are looking at compounds that can prevent the virus from making copies of itself. In the second, we're looking at anti-inflammatories. And so part of the reason we think that there's severe disease following coronavirus infection is not necessarily due to the virus continuing to make copies of itself. It's the immune response going down a bad path, or a pathway that winds up actually causing more disease symptoms rather than preventing them. And so this category of treatment options is called anti-inflammatories, and it's drugs that can help kind of dampen some of these immune responses that are more dangerous or could potentially lead to more severe disease outcomes. And then the final treatment option is called antibody treatments. And so antibodies can recognize the virus and bind to it and either prevent it from getting into the next round of cells and or target it to different parts of the immune response that can then destroy the virus. And so similar to vaccines, and that's basically what this slide was originally generated for, both the treatment options as well as vaccines have to go through clinical trials, evaluations in people to make sure that they're safe before they can be used. And so I wanted to kind of walk through this again. So in phase 1, you use the smallest number of you use the smallest number of volunteers. And researchers are basically trying to say, you know, are these treatments safe? Are there any serious side effects with the treatment? And so basically, you know, is there any reason not to try this drug in people who are sick? When you move to phase 2, these drugs are tested in kind of several hundred volunteers, some of which may actually be infected with what you're trying to prevent. And so then you're looking at more short-term side effects. How does the body respond to the compound? Are there any or does the body actually respond to the compound? Or are there any protective outcomes? Does this appear to be stopping the virus from making copies of itself or dampening the immune response? In phase 3, you're looking at the largest number of volunteers, and you're basically just trying to figure out, you know, does this compound do what you need it to do to prevent the disease severity or stop the virus from being able to replicate, copy itself? You know, are there any other outcomes that we should be looking at? And so right now, because we're in a global health crisis in the United States, the Federal Food and Drug Administration has the ability in a public health crisis to use emergency use authorization. So it could kind of bring treatment options or different vaccine candidates to the general public a little more rapidly than it ordinarily would, to be able to offer alleviation of some of these public health issues. So I just wanted to make sure that people understood that. And then phase 4 is full-out treatment approval by the FDA. So right now, what is available on the market, and rather than show you the very long and complex scientific names for some of these treatment options, I basically just categorized them in the 3 different categories that I showed you earlier. So treatment option A is an antiviral that's approved for use in the United States. Treatment option B is convalescent sera or antibody derived from a patient, and this is currently available for emergency use authorization. And C is a synthetic antibody that was generated by a company that's targeting COVID-19. This again is approved currently for emergency use authorization. Treatment option D is an antiviral in combination with an anti-inflammatory, so something to dampen some of the negative disease outcomes. And this is also approved for emergency use authorization. And then treatment option E is actually a combination of 2 different antibody treatments, which is again also approved for emergency use authorization. So more and more treatment options are coming online. So when will even more be available? Basically, I found this very interesting table where this group is in essence basically tracking all clinical trials of different compounds and tracking where they are. So everything that I'm mentioning in this particular slide is either in the 2nd or the 3rd phase of clinical trials. There are currently 4 antivirals that are under investigation, 16 anti-inflammatories, so those that are going to dampen the bad disease causing immune responses, and another 12 antibody treatments, and some of these may actually be in combination. These are the most up-to-date numbers for both January and February, and so I think there'll probably be even more coming online. So we are getting to a point where we're going to be able to offer treatment options for those who are infected. But I also do want to say, as much as possible and in consultation, obviously, with your healthcare provider, if you can get vaccinated, please do. That will, I think, be one of the major things to get us closer to our old normal as quickly as possible. So now, finally, and what happens next, and how do we prepare for the future? So scientists and medical professionals are continuing to learn more about the virus and ways to treat patients and prevent infections. This has all still been very new. I know everybody likes everything to happen relatively quickly, but sometimes science just takes time, and we have to take the time to learn how to do the best things to prevent disease symptoms and prevent further infection. Things you can continue to do in the short term, and I know everyone's tired of hearing it, but they really do work. Social distancing, as well as wearing masks effectively in public. You just have to fight through it and make sure that your nose and mouth are covered. I know that's really challenging. And also just, good old fashioned washing your hands very often. This is really a really great way to prevent disease spread for sure. And things everyone can do in the long term include, wearing a mask and staying away from others if you're sick. I'm kind of hoping, and it may be idealistic, that maybe culturally we can move a little bit closer to what Asia does, and that is that if you're sick and you have to leave the house, that you just don a mask, and that way you're not spreading whatever it is that you have to other people. And so, I think that's kind of where we are. Get the vaccine if you possibly can. Be patient. They're getting things out as quickly as they can. Vaccines typically take at least 10 years to come online. These have come online in a year. And so it's really an amazing achievement to be at this point where we are, even as hard as it's been. So that wraps up my talk. So next week, we will actually discuss exposure and infection, those portions downstream of transmission. So that will be very exciting. And then we'll move on to the coming events.
Yeah, great. Thank you, Amy. Thank you very much for that presentation, taking your time to discuss the topic and everything that you know about it. I know everyone in the audience appreciates it. As you were just mentioning, do want to put in a plug for the upcoming seminars in this series. So every Tuesday in March, next Tuesday, the 16th, we'll hear from Dr. Katrina Waters. The following Tuesday, we'll hear from Dr. Kristen Omberg. And I think these will be great talks as well. So we're going to jump into the Q&A session here in a moment. We have 235 people that joined us tonight, so another great turnout. I guess I'll have to pound the pavement. I think we had 250 last week, but still wonderful to see so many people here. As we mentioned at the top, we have a panel of community representatives that we'd like to start with. So we're going to allow them to each ask a question. I encourage everyone listening, please continue to type in questions in the Zoom chat. We've been collecting those, and we'll be getting to those after our panel. So we'll go in the order of the introductions from the beginning. We'll start with Martine Valadez. Martine, do you have any question? Do you have a question for Amy?
Yeah, so there's some evidence that certain groups, Latinos and African Americans, have higher rates. So some of that has been attributed to work and things like that. For example, here that individuals work in the fields or in packing houses, etc. But is there any evidence that ethnicity, if there's any evidence that ethnicity plays a role in either transmission or the severity of the disease?
I am not aware of any that there are, but that's a fantastic question. And I'll be happy to do a little bit of digging. I will make a note to myself. I am not aware of any right now, but no, I have heard what you stated as true. But no, that's a great question. Thank you.
Thank you.
Great, thanks, Martine. We'll next take a question from Justin Rafa.
There was so much attention early on in our COVID response to surface contamination issues and the importance of hand washing, sanitizing doorknobs, tabletops, and other surface areas. Now that we better understand the transmission nature of the virus as being largely airborne due to respiratory droplets, to what end should we continue to prioritize surface sanitation efforts? And I ask this knowing that many businesses and schools seem primarily focused on safety protocols around sanitation, with less emphasis on indoor air quality, circulation, and the physical makeup of their facilities.
I think scientifically what you're saying is true, that it's less likely to be spread on surfaces and more likely to be spread via airborne routes. And so I think, focusing on the air is probably a good thing. But I also think it's much easier for people to wrap their brains around surface decontamination. So I think it's a good place to focus. It's not a bad thing to just do in general. Washing your hands more is a fantastic thing to do. It's going to help you overall. And so I think that may be it. It's probably easier and less expensive to focus on those as well. So yeah, I mean, I think from a true transmission standpoint, I think this virus is very airborne, and that's most likely where we're going to be seeing it. But it can actually hang around on surfaces. So it's not wasted time, but I think a little bit of focus on the air is good as well. And I don't know how to help people do that. Thank you for your question.
Yes, thank you. We'll move on to Kate McAteer.
Thank you, Amy. That was a wonderful presentation. So Justin mentioned schools, and I think, there's a lot of people that are concerned about kids and students and the adverse social effects that this might be having on them not being in school. But with the increasing number of cases, I think you mentioned over 2,000 cases now of the multisystem inflammatory syndrome in children. And thinking that, clinical trials are done in adults, are there treatments for MIS in kids or?
When I saw today, they did not list any treatment options. I think it's basically just kind of, it's inflammation of different organs is what's happening. And so I think that they're just like treatments of symptoms, but not necessarily a way to stop it outright. At least scientifically, that's what I could come up with. So I know that's really challenging, and we don't have any good answers. And that's definitely something that's come out of this pandemic that we had never seen scientifically before. We really thought that anyone under the age of 19 was in essence safe. So that's something that's really been unexpected. And so I don't think that we have good options right now. But that's 2,000 cases out of the United States at large. And so, yeah, I don't know how to move forward in that, but I'm hoping that we will have better answers soon.
Thank you.
Thank you. And we'll round out the community panel with a question from Loanne Ayers.
I have a question regarding pre-existing conditions. Obesity is one of the key pre-existing conditions that impacts severity. And in our community, 1/3 of adults and 1/3 of teens are obese or overweight. And that was pre-COVID. So many of those who are struggling with obesity, there's also a strong tie with poverty. And our nonprofits serve many people who are in poverty and struggling with nutrition insufficiency. What key message points about nutrition or, other than just lose weight, can we convey to those who are struggling with this potential pre-existing condition that would help with protective factors or recovery?
That's a fantastic question. And, I am really a basic science virologist, but I mean, if there's any way to exercise, take advantage of, having the ability to walk during the day or, anything like that, low cost. But basically, being overweight winds up affecting your immune response. As non-intuitive as that is, it really does affect how your body responds to things. It's more stress, those sorts of ideas. And so anything that you can do to de-stress, which is super difficult, as well as exercise more. I'm not trying to say lose weight, but do something, because exercise of any kind is actually going to help you move closer towards a more healthy outcome. But you have a very tough job, and I don't know what the appropriate response is. But that's the comorbidities are super hard, and we just don't have it all figured out yet. But hopefully, maybe we'll learn some more answers that will be helpful. But no, fantastic question. Thank you.
Well, stay tuned, Amy. Thanks.
Absolutely, please do.
Thank you all. Those were really great questions. We have a lot of additional really great questions coming in through the Zoom chat. So I'd like to start bouncing some of those off you, Amy.
Okay.
Let's start off with, if bats have a reduced immune system, why don't they succumb to infections more often, or do they?
That's a great question. So I think they probably have what, the immune response that they need to protect themselves, but what it does is allow them to kind of have other things replicate. unchecked kind of in the background. So it's not that they're missing key components per se, but they just don't maybe have kind of the overreaction that sometimes we do that causes some of the disease symptoms. So maybe it's kind of like a slightly different way to look at it. I think that they have the immune response that they need to have evolved and still be surviving in a very diverse species. So maybe think of it more as a dampening as opposed to missing key components. Hopefully that makes sense. Uh-oh.
Sorry about that. Sure, thank you.
Excellent.
Okay, there's several questions related to the portion of your talk talking about viral mutations. So
Okay.
let's start. How much of an impact does larger portions of the population becoming infected have on the rate of mutations?
It will drive it up for sure. So the more people that are infected, the more mutations that could potentially come out. I mean, more people infected, more baby viruses, more potential for different types of mutations. So it definitely will drive it up. And that's actually why, I mean, you know, with only 8,000 cases with SARS coronavirus 1, which was more than plenty, and only 2,500 with MERS, we really haven't had a chance to study this virus as it evolves in people in a huge number like we certainly have now. So I think we're going to learn a tremendous amount. But as I keep saying, everything takes time. And unfortunately, that's one thing that we're in short supply of right now. So great question. Thank you.
Sure. Kind of going along that same line of thinking, is it possible that multiple genomic variants of the virus can be present in the same person?
I think it would potentially be possible to be infected with different variants. Yes.
Okay. And if mutations are, as you explained, really just mistakes in the viral replication process, in order for there to be common mutations that are popping up, like the UK variant, is that just a common mutation, a common mistake that's happening all over the place, or is it actually a beneficial mutation that's then allowing the viruses to replicate more readily?
So it would kind of be like an additive effect. So you would have one mutation that would allow it to pass on, and then it would get another one that would also be beneficial and kind of help that guy move forward more than the others. So I think it's kind of I think of it as kind of like a stacking effect that there are mutations that, you know, if they make changes here, then potentially it can move into people faster. So it would infect 3 people as opposed to just, you know, this other virus that was kind of lagging along could only infect one person in that same amount of time. So that the 3 people virus is going to, you know, exponentially grow faster. And so hopefully that answers your question.
I think that does. Yep. Can you briefly touch on similarities or differences between some of the major variants of the virus that are circulating right now, and any thoughts on whether current vaccines are effective against different variants or just the original one, or is that still TBD?
So we're still learning, obviously, about how well the vaccines, you know, how well the vaccines protect overall and how long it will last. But the reality is that the vaccines work even better than we need them to. So we think that these new variants will kind of fall underneath that umbrella. It may not work quite as well, but it will protect you and, you know, have you replicate less virus, get less sick, and things like that. And so the current thought right now is that, you know, overall the vaccines that we have should still protect us against these new variants. And I actually pulled up the CDC website before I was speaking today, and I think in the United States there's still only about 3,300 cases with the 3 new variants overall. So I think there's a lot of attention, but maybe not quite as far spread. I mean, like 2 of them have under 100 cases each. One of them has over 3,000. So I don't think it's quite as widespread as some of the attention would potentially indicate. But just, you know, something to keep in mind, that I think that the current vaccines should work against these, at least as far as we know right now.
Great, thank you. Couple of questions in the realm of species hopping, species transmission. So there have been reports of lions and gorillas at zoos being infected with with SARS-CoV-2. How unusual is that for this much species hopping? And do you think that has implications for future infections, future zoonotic infections?
I think, you know, it's hard to know, and it's probably virus dependent, you know. So each virus is kind of in its own little category, and we just don't know. I saw the cases of cats being infected. I know dogs can be infected as well. So the gorilla, not as surprising, because it would have a more human-esque way for the virus to get into the cell. maybe for whatever reason, this particular virus, the cat receptor is very similar to human as well. Like I said, it can be very species specific. Mouse can't get infected, but a hamster can. So, it's a little hit or miss, and it just depends on how well those proteins can kind of mesh the handle for the cell and the handle for the virus. So, I think kind of all bets are off at this point. As much as the virus has been able to replicate, there are lots of different variants out there, and so one may prefer cats, over humans, honestly. So, not too surprising, but you just never know what you're going to get.
Okay, great, thanks. There was a question in the chat that also I had as I was listening along. So I think if I understood correctly on your slides, the common cold made the jump from cows to humans in the 1800s. Is that
one of the coronavirus strains that causes the common cold? Yes.
Okay.
We think
so.
Yes.
Explorers
not the common cold. So the common cold is actually caused by several different viruses. Rhinoviruses can cause it. Coronaviruses can cause it. Adenoviruses can cause it. So I think we as humans have kind of collectively grouped symptoms together, but they can actually have many different causes. And so one of the coronavirus causes of the common cold, we think, kind of came from humans' close interaction with cows.
Okay. So pre-1800. Yeah, it does. And so pre-1800s folks were not completely common cold free then?
No.
Excellent. Okay. Could you talk a little bit more about the evidence we have to understand that bats are the original reservoir, why that's known?
So scientists have actually basically been taking bat poo, believe it or not, and extracting nucleic acids out of that and learning about the genomes of things that are inside of bats. And basically what scientists have found is that there are lots of different coronaviruses. They're general bat coronaviruses, and some of them were very similar to SARS coronavirus, the first that was identified. And we found others that are more closely related to MERS coronavirus. We found others that are closely related to SARS coronavirus too. So the current thought process is that that's the most likely reservoir. Now, whether or not, like I said, there was an intermediate animal required, like the camel for MERS coronavirus, it's hard to tell, but that's really how we've figured that out. So we're trying to just kind of do general surveillance to look and see, what kind of viruses are in bats, to see what they could be carrying and potentially what we need to be kind of on the lookout for. But that's kind of how that's done. So hopefully that will continue for sure.
Great. Okay. I think that kind of leads nicely to another question related to the quickness with which the current vaccines were developed. Do you think that this has obviously gone faster than many people might have expected? So do you think that there were some, potential for oversights or things that could be leading to long-term health effects from those vaccines, or?
So basically the vaccines went through the exact same stages that any vaccine would go through. They just went through it in a condensed time frame. So all the same safety metrics, all the same evaluations were done. They were just done on smaller populations of people in a shorter amount of time. And so I think, to the best of our ability, science has provided us a way to protect ourselves from the virus. So hopefully not, but, viruses that or vaccines that took, many more years have come on the market. There have been adverse reactions sometimes, and they've had to be removed. So I feel like, to the best of our ability in a global health crisis, the FDA has approved the best, for emergency use, the best that it can, and what we have out there. So, everywhere that you look, that's basically, it tells you that they've been evaluated in the same manner as any other vaccine would. It's just in a shorter time frame.
Okay. Couple more, I'll put you on the spot with a couple more vaccine related questions. So once we get past the initial phase of vaccine scarcity and toughness to schedule and get your appointments, would there be a benefit to taking having multiple types of vaccines? Would you get better protection if you had both an mRNA and a J&J version?
Not that I know of, and right now, the definite recommendation is that if you get the first dose of Moderna, to get a second dose of Moderna, or same with Pfizer, get both doses of Pfizer. I'm not sure I see any kind of additive, or that I know of an additive effect for mixing the vaccines. I think get the 2 doses of Moderna or Pfizer, or get the single dose of the Janssen or Johnson & Johnson. And I think that should do you, at least for now. And I know a lot of people really want to know how long does it work for, and the answer is we just don't know because there hasn't been time. And so, you know, as time progresses, we'll have a better idea of how long people are protected by the vaccines. Hopefully, we won't have to get, you know, boosters very often if we need them at all, but only time will tell that, unfortunately.
Okay, and you hear a range of severity of reactions to vaccines. So does the level of side effects that one might have correlate with the level of immunity they're gaining from it?
I don't think so. I think it's just how your body's immune response reacts. I think that's the thing that's maybe harder for the general public to realize, is that, you know, it seems like you were basically having COVID-19 and your reaction, but it's basically just your immune response reacting to this one little piece of the virus, and saying, hey, I know about you, I'm going to take you out now. You don't feel very great, but, you know, it's what you want to have happen, because, you know, it means that your immune response is definitely recognizing and kind of jumping on it and taking care of it quickly. I think everybody reacts to things slightly differently, so I wouldn't be worried, you know, if you didn't have very many symptoms, you know, after your second vaccination, and you had somebody who did, I wouldn't necessarily think that those were correlations between how well you're protected. It's just probably how your body reacts, you know, as opposed to how theirs does.
Great, thank you. I think we have time for a couple more questions here. So you mentioned that viruses lack proofreading functions when they're replicating their genetic material. Is that true for all viruses? Do some actually have proofreading activity?
Actually, some do. Coronaviruses, believe it or not, they actually have a proofreading enzyme, but they still make mistakes. They just make it at a lower rate than you would typically expect given the size of the genome. And that was actually a huge change in dogma. So coronaviruses are actually the largest of their type of virus, which basically their genomes are like a big piece of message-sense RNA, if you remember back to, you know, high school or college biology, which means it can be translated directly into protein. And so it turns out that there actually is one protein within the coronavirus genome that does do proofreading. Obviously, it's not perfect either, but it keeps the mutation rate much lower than you would expect for the size of genome that coronaviruses have. But no, fantastic question.
Great. And just as we talked about mutations being able to make the virus more transmissible, is there equal opportunity that mutations can happen that impact the virus's ability to replicate and transmit?
Absolutely.
And if so, just, you know, is it possible that we might see a decrease in cases due to those sort of mutations?
I think that those viruses are just going to kind of fall away. I'm not sure. The virus is not going to seek to evolve to off itself. It's not a living organism, but, you know, it does still want to make more copies of itself as much as possible. And so I think, you know, the things that are going to move on are ones that do that better, not, you know, obviously ones that do it worse. And so I think really the thing that's going to protect us is not necessarily the virus kind of evolving itself off to not be able to infect very well, but to protect ourselves, you know, with vaccinations, and give the virus nowhere new to infect. So I think that's probably the best that we can do. Unfortunately, they don't tend to evolve themselves into not existing anymore, but a great question. No, very good thought.
Excellent, thank you. I think we're gonna stop there. We're just coming towards the top of the hour. So really wanted to thank you again, Amy, for taking the time to share your knowledge here tonight with everyone. Also wanted to thank everybody in the audience that attended, and a special thank you to Justin, Martine, Kate, and Loanne as our community representatives. Really appreciate you guys being here, fitting this in with everyone's tight schedules. One final reminder that this is talk 2 of 5, so there'll be 3 more seminars every Tuesday here in March. There's links in the Zoom chat that have been posted to the PNNL events page, where you can find more information about those talks and other upcoming CST seminars. Again, would like to remind you that as you close out today, you should have a link popping up to a brief survey. That would be great if you could participate in that. And yeah, with that, we'll take off. Join us next week, March 16th. Katrina Waters will be presenting Behind the Mask, the Science of Stopping the Spread, 5 p.m. here on Zoom. Look forward to seeing everyone again. Thank you.